Welcome to session 7. We have got two fantastic speakers lined up, and the first one here online is Thom Sharp. Unfortunately, Thom can't be with us today in person because he's not feeling very well. Thanks for making it anyway virtually, Thom. As a quick introduction to your background, Thom did his PhD in Bristol, joined with Paul Verkade and Dirk Wolfson. Then he moved on as a postdoc to Struby at Oxford. And then he went abroad to the Netherlands for a while, where he did a second postdoc and then became assistant professor and then later associate professor in 2021. And then he decided he wanted to go back to his origins. So in 2024, he returned to the University of Bristol, where he now works with the same people who did his PhD with, which is great fun. And we are very happy to have Thom down in the southwest of the UK with us. And yeah, without further ado, stage is yours, Thom.
Thank you very much, Bertram. Thanks for the invite and the introduction. So yeah, hi, good afternoon, everyone. It's a real shame I'm not there in person. I was really looking forward to being there and meeting up with you all. But I'd like to thank the organisers and the tech team for the last minute changes of plan for me to be able to present to you online instead. So as Bertram said, I moved back to the UK after running a group in the Netherlands for more than 11 years. And it's a real pleasure to be back at my alma mater and of course to have the opportunity to present to you today. Now, what you can see here are a bunch of proteins in the process of activating the human immune system. Now, we've imaged these proteins as the purpose of my lab is to understand and eventually control the human immune system. But of course, to understand it, you need to be able to see it. Now, imaging purified components can only get your understanding so far. To really learn how we can control immunity, we need to see it in action. And that, of course, requires in situ structural biology. I'm going to tell you today about how we do this and what we've done with structures such as these. Now, as I said, my lab is interested in the human immune system, many aspects of it. But today I'll focus on part of our immune system called complement. Now, complement plays a key role in immune defense against invading pathogens. But when complement goes wrong, it can lead to things like autoimmune diseases like lupus or rheumatoid arthritis. But conversely, complement is also a major effector function of antibody immunotherapeutics, which are used to treat things like cancers and various other diseases. Now, many of these components circulate in your blood and tissues in an inactive form. Now, the classical complement cascade can be activated when certain antibodies bind to antigens, and this might be on a bacterial cell surface, as shown here, and they form a platform. And this platform acts like a landing pad for the first component of complement called the C1 complex. Now, C1 comprises a ligand binding and scaffold complex called C1q, shown here in blue, and two serine proteases, C1r and S, shown here in purple and pink, and these form a heterotetramer inside of C1q. Now, binding of C1q to these antibodies or other ligands induces activation of C1r and S, and this initiates a cascade of proteolytic events, resulting in opsonization of the bacterial membrane. Essentially, the membrane is covered with proteins, and these proteins act like seasoning for macrophages and neutrophils. They want to come along and phagocytose or eat this cell. And while the phagocytes are on their way, complement is busy destroying the bacterium. The cascade terminates with the formation of a pore called the membrane attack complex, which lyses the bacterium. So if it's not been eaten by macrophages, it will be popped by this macpore. Now, we've spent a lot of time studying antibody-mediated complement activation and have used cryo-electron tomography to solve the first structures of these initiating complexes with IgG1, IgG3, IgM, the terminal macpore, and a bunch of other important complexes as well. Now, most of these structures, all the ones with a green envelope, have been determined in their native environment. That's human serum, which means everything that's in your blood, except the red blood cells and clotting factors, is still there when we image it. These are also flexible proteins. They have to be in order to do their job. Nevertheless, we can see them. Now, whilst we do routinely solve proteins to sub-2 angstrom resolution, sometimes low-resolution maps contain amazing information too. This is a 22 angstrom resolution map. It was the first complete structure of an antibody bound to surface antigens. So here we can see IgM in green, C1q in blue, the proteases C1r, in purple and C1 pink. And this C1s, sorry, C1s in pink bends down to this cyan colored protein. Now, this is called C4b. C4b is actually the product of this enzymatic reaction. And what this means is in unpurified human serum, we could actually catch this enzyme in the act of initiating immune defense. Now, we've done the same for a bunch of different proteins and learned loads about how antibodies in complement function, modulate, perhaps don't always work. And I won't have time to talk about that today, all the things we discovered. Instead, I'll zoom into just one example where these kind of in situ data were crucial. Um, so this kind of in situ structural biology, when we're looking at the human immune system being activated in human serum on these antigenic surfaces. This allowed us to compare all of these similar but different structures of antibody complexes caught in the act of activating the classical complement cascade. There were quite a few structures of purified components as well. And what this allowed us to do is see what changed between the solution phase or inactive complexes and these antigen bound or active complexes. And we discovered that the binding interfaces between antibodies and C1q are actually just these tiny little peptide domains on the periphery of the FC platform. So these tiny little peptidic domains are on the periphery of these antibody platforms. So what we did was design and synthesize a peptide comprising only these amino acids. So this is what it looked like in our low resolution structures. We could essentially build these from other PDB models as well. So what we did, we took this little bit that was interacting with C1q and cyclized it so that it made the conformation that was found in a whole antibody. Now, this is work done by my PhD student, Sebastian, who's actually graduating next week, and a long-term collaborator of mine, Amy Boyle. So when we took this cyclic peptide and conjugated it to a lipid, or a lipid membrane rather, and added human serum, we found that the peptidic interfaces on their own were sufficient to activate complement. And that's what you can see here. So there's no antibodies, there's no other ligands there that are going to activate complement apart from our membrane-associated ligands. Now, what's rather cool is these little peptides are only 1 kilodalton in mass compared to a megadalton in mass for an IgM complex. So we've been able to distill the complement activating ability of an antibody into something that's 0.1% the mass of an antibody, i.e., we've got rid of 99.9% of it. Now, we've called these peptides peptactins for peptide-based activators. Now, this first generation of peptactins, they weren't very good, but they did show us that the concept worked. So we then used a technique called rapid selection, which we did with Senyo Jonkes at the Free University of Amsterdam, to evolve our minimal motif into a more potent molecule. And now we have what we term second generation peptactin. So you can see there's certain motifs here. There's PXP motif, PSP, and it's now PDP just here. So these second generation peptactins can actually recruit specifically C1 from human serum. They can activate the C1R and C1s serine proteases and even induce MAC-pore formation and lysis of cell-like membranes. So now we're ready to apply these. When we target these peptactins to lipid membranes by conjugating them to a lipid with a bunch of different techniques, we've done it quite a few times now, they behave pretty much identically to antibodies. So now we've got these kind of drug candidates, what are we going to do with them? Well, these peptactins, they're small peptides and they can essentially be tagged onto almost anything. And that allows us to install the ability to activate complement into any molecule that we like. So this is a whole antibody. These are antibody fragments, Fabs, nanobodies, little peptides. These three on their own don't have the ability to activate complement or the immune system. Now we can stick on our little peptide, our peptactin, and give these targeting molecules the ability to activate our human immune system. So we're working up a few targets at the moment. One is against antibiotic-resistant bacteria. We're looking at targeting B cells. These divide uncontrollably in lymphomas and leukemias. So we can use our innate immune system to kill those. B cells also sometimes erroneously make antibodies to perfectly innocuous targets. These are called autoantibodies. That's where you get autoimmune diseases from. So we're looking into killing only the disease-causing population of B cells as well. But the reason I told you all of that is because this whole thing, we're now spinning out a company as well. This whole thing was made possible only when we image these molecules in their native state. Now, I mean, you're the kind of target audience where I don't have to really convince you of this, but if any of you are in doubt, hopefully I've convinced you that imaging proteins in their native environments really helps, indeed is essential for us to learn more about how our immune system actually functions. So obviously we want to move towards even more native-like systems, whole cells. And so I'm now going to tell you about some of the methods we're developing to move our research towards cellular systems. Now, this is work performed by two of my PhD students, Martin Tautel and Mart Last. Martin's now a postdoc at Max Planck Institute, and Mart has literally just started his postdoc at the LMB in Cambridge, and a long-term collaborator of mine called Leonard Vortman, an amazing light microscopist. It's worth saying everything I'm going to talk about now is open source. It's all published, code, CAD plans, everything like that. We wrote a couple of back-to-back methods chapters last year on how to build what I'm about to tell you and how to use it as well. So see this as a little bit of an advert as well. But first, I want to really put this in context. Why do we want to do this? So a single one of our cells is around 20 microns wide. But an E. coli bacterium is only 2 microns wide. It's pretty much the smallest organism you can see with conventional light microscopy. But viruses, of course, are 200 times smaller than our cells, and most proteins are 10 times or more smaller than that. In fact, a single antibody protein is 2,000 times smaller than our cells. Now, this movie is to scale. You can see that proteins are tiny, but electron microscopy, we can see them. Cryo-EM allows us to see their constituent atoms. So now we have a different problem. Even though we can see them, how do we find these tiny proteins? Well, by using biochemistry, we can add a fluorescent beacon to our molecule of interest. It's guiding us where to look. This is one of the advantages of fluorescence light microscopy. We can combine this with electron microscopy to form correlative light and electron microscopy, or CLEM. And now we've pioneered several methods to do so-called cryo-CLEM. But now we've combined this with super resolution cryo light microscopy as well to form, we need a better name for this, so if anyone's got any ideas, super resolution cryoclem or SR cryoclem. And this allows us to find individual proteins with a 30 nanometer accuracy or precision. Now, there are a few methods to perform super resolution light microscopy. The most applicable to cryoclem is single molecule localization microscopy. This includes techniques like PALM or STORM and delivers generally the highest resolution. So here's a simulated image of what a sample might look like in a conventional microscope. To do single molecule localization microscopy, essentially we activate a small subset of spatially separated fluorophores. So here's just one fluorophore. We can then find its center and plot it with very high accuracy. We can then turn that fluorophore off and turn on another fluorophore and again find its center and plot it with high accuracy. Now, as long as the fluorophores are spatially separated, we can find their centers, do this again and again, and get a much higher resolution than conventional fluorescence light microscopy. Now, VIDI was just the name of the funding agency from the Dutch government that awarded me a grant to develop this cryo super resolution technique. But very quickly, we found, perhaps unsurprisingly, that photons devitrify the ice, and this completely ruins cryo-EM imaging. But we found that devitrification is dominated by the laser intensity and not the total dose or the duration of imaging. Obviously, that's how it works in cryo-EM. It's the total number of electrons which reduces your resolution. Here, that's not the case. It's the fluence. In fact, what we learned is we could avoid devitrification entirely. Here it says for 30 minutes. We've done it for much longer. As long as we stay below a certain threshold, in this case, it's 28.5 watts per square centimeter, we see no devitrification at all. Doesn't matter how long we image for. But if we increase that intensity just a little bit to 33.5 watts per square centimeter, devitrification occurs rapidly. Within 5 minutes, Marta's now shown that it occurs pretty much instantaneously. This was a consistent phenomenon with devitrification starting rapidly in the center of the grid squares and moving out from there. However, Martin had found that this is dependent on the kind of grids you use. And we did a bunch of data collections on a huge number of different samples and grid supports and things. And all these data allowed us to construct a model of the heating. And we found this was best explained by the copper grid bars acting like a heat sink. And this then quite obviously opens a door to identifying and maybe developing better grid supports. But we also took the very pragmatic approach. So Mart essentially bought up all of the commercially available grids that could be used and screened them all, trying to identify, understand, and therefore mitigate this heating effect. Now, the best ones we've found are ultrafoils, gold grids with a gold film on top. These offered very good heat characteristics. We could use a laser intensity 5 times what we could get away with before. They aren't perfect, however, but we do know how to make them perfect. So please get in touch if you're interested in working with us to develop these kind of perfect supports. Now, these two above, this Formvar, which is kind of plastic and silicon monoxide, which is basically glass, we couldn't devitrify the samples. We maxed out our lasers and we saw no devitrification at all. Now, this made us very excited, but it turns out that these support films also spontaneously blink. So even if we don't have any support films there, our films, if we don't have any sample there, if we don't have any fluorescent protein there, our support films are spontaneously blinking, which is obviously a massive problem when you're trying to find individual fluorophores. But maybe these would be interest to people doing things like scanning microscopy, like STED or Resolved or something. But we essentially only use ultrafoils now. Obviously, these are biologically compatible as well, which is very helpful. Now, obviously, the next step is just because you don't see devitrification, is it still a problem? Super resolution imaging does not damage protein structures. So here's apoferritin being imaged before and after being exposed to 3.6 million photons per square angstrom, and the resolution remains unaffected compared to without the photons. We can still see through the holes in tyrosine side chains and things like that, which is very reassuring. Now, for fluorescence light microscopy, we obviously need fluorescent beacons. Now, traditionally, we'll use fluorescent proteins such as GFP. However, GFP is not photo switchable. For super resolution light microscopy, for this single molecule localization microscopy, we need photo switchable proteins that can be turned on and off with different wavelengths of light, such as RSE GFP2 shown here. So here you can see an increase in the fluorescence after an activating pulse of 405 nanometer light, and then illumination with 488 nanometer light is a deactivating light. That then rapidly turns off the RSE GFP2 before we then repeat the workflow. And each of these is essentially when we turn off a different, hopefully spatially separated subset of fluorophores. Now compare this with GFP and you can see there's no photo switching. And we discovered a few years back now something rather fascinating. Fluorescent proteins behave differently when prepared for cryo-EM. Not just when cold, they are vitrified just like for cryo-EM. Even non-switchable proteins like GFP become photo-switchable when prepared for cryo. And this means they can be used for super resolution cryo light microscopy or super resolution cryochem. And this was true of all of the green fluorescent proteins we tried. Non-switchable GFP becomes photo-switchable. Photoactivatable GFP becomes photo-switchable. Fast and slow switches become photo-switchable. Padron is what's called a reversible, a positive switcher instead of a negative switcher. It turns off and on instead of on and off. Again, becomes photo-switchable. Even photoconvertible proteins that go from one color to another, like MRSFP, become photo-switchable when prepared for cryo-EM. And these results mean that any fluorescent protein here in this list can be used for super resolution cryo light microscopy. This is nice because it means there's no need to reclone specific variants. So even though all of them are photo switchable, they weren't all as good at it. We picked the protein with the highest dynamic range. This was RSE GFP2 and used it to label intracellular microtubules. So here's a cryo light microscopy image of microtubules inside a cell labeled with RSE GFP2. And if we zoom in just here, we can see the diffraction limited cryo light microscopy image. And we can compare this with the super resolved image, which really, I think, shows you the power of this technique. And we can correlate this super resolution light microscopy image with cryotomograms of the same sample, which we can combine with structure determination using subtomogram averaging to solve structures of proteins in situ. So we've actually written some software to make this whole process semi-automated called SC Nodes. It's available on GitHub and in Nature Methods. This was largely written by, or entirely written really by Mart. Um, Now, what SC Nodes allows you to do is rather quickly and intuitively play with various settings. Now, if you ever use Blender, you'll be familiar with these kind of nodes that you can move around. And these essentially let you play around with settings, and it spits out various parameters and statistics telling you if you're doing a good job or a bad job. Things like alignment of the frames, background subtraction, finding the centers of particles, etc. Um, we find it very, very easy to use. Um, Within SC nodes, you can also correlate all your 2D and 3D data. So after you've got it correlated, you can then, including like tomograms and things, you can align your tomograms to the super resolution reconstructions. And then you can play with each individual part of your data. You can go back and redo your super resolution, see if you can get it closer to what you now know is the ground truth to give you better settings and things like that. And it all remains aligned. And what this all means, the reason I wanted this software is because we can then do fluorescence guided particle picking. So we're guided by the super resolution. So here you can see some, it's rather faint and faded here, but you can see a little fluorescent spot here. And now inside of this, there's a protein called TAP1. We have no idea what TAP1 looks like, but now we can go hunting within these fluorescent spots for new repetitive looking structures and pick these out of our tomograms. But we do want to go further. Essentially, we want to take images like this and turn them into this, right? Rich in contextual and macromolecular detail. Now, so far, our developments in SuperRes CryoClem has allowed us to color in one molecule at a time with around 30 nanometer precision. But now we're extending this using multicolor cryo super resolution imaging. So we have tested a bunch of different colored fluorescent proteins and identified one RS tag RFP that is compatible with our current best green probe. Now, this is work in progress, but I'm very hopeful you'll be able to read about it soon. Now, another way of getting this kind of information is with segmentation. So Mart's written two more software packages. He really went on a splurge of software writing towards the end of his PhD to automatically recognize features and regions of the cell to provide this ontological and macromolecular detail we need for in situ structural biology. Now, one of these software programs is called ICE, which does a fantastic job of machine learning based segmentation to automate particle picking. What it's really great at doing this in a context aware way. Essentially, models can fight against each other based on what is around, and this allows you to tune the models really nicely. Another package is called POM, which we released on bioRxiv a few months ago now. This can now use the context of segmented particles to generate ontologies. Essentially, it can recognize cellular features, not just macromolecules based on what's around. What this is really exciting. What I wanted was essentially to turn cryo-electron tomograms into searchable databases. I mean, what I really want is a single piece of software where you can automatically reconstruct and segment your tomograms. Essentially, you set your microscope running, you come back when it's done and you type in, find me all the tomograms where a microtubule is within 10 nanometers of a membrane, for instance, if you're hunting for membrane proteins. And then it provides you with all the relevant details, and you can then ignore the other tomograms that perhaps don't contain motor proteins. Hopefully, this will also improve data sharing and reuse. Mart recently deposited a massive database of segmentations into the CZI's CryoET data portal. Now, just to finish off with an example of how we're using all of this, as I told you at the start, antibodies and complement components mediate cell clearance using phagocytosis. So we can take primary macrophages and differentiate these, primary monocytes, sorry, and differentiate them into macrophages, gives us a lot of very nice genetic control over it all. We can then feed them opsonized material that we have chemical control over. We can make these synthetically. And when we feed the macrophages, we see this beautiful phagocytic process taking place. Now, although we have some structured purified components, these don't tell us how this works or why indeed it sometimes doesn't when it should. Now, rather excitingly, we're getting a cryo-FibSem in Bristol. Well, it's already here. It's in boxes at the moment. It's being installed at the moment, which we can then use to thin samples. So we can look at very early stage of phagocytosis, very late stage of phagocytosis, and of course, a bunch of other biological and immunological problems. So biochemically, we have a list, many lists of proteins and receptors involved in phagocytosis. But immunologically, we don't know how they orchestrate the process of phagocytosis. There are no structural data of any of the complete macromolecular complexes formed. So guided by cryo-superres, we can then zoom in with cryo-ET to image proteins and receptors specifically found to identify the molecular events of phagocytosis. And that'll allow us to replace these kind of textbook images where you just have blobs engulfing a protein with the real structures of these molecules. Essentially, what we want to do is combine these nanoscale structural biology techniques to understand and control the macroscopic biological effects of immune defense. So some conclusions. Cryo-ET provides function information by solving in situ structures. We've solved a bunch of these caught in the act of initiating immune defense. Now, these in situ structures provide contextual information, very important, that explain how the immune system is activated, modulated, and inhibited. We're using these structures in a huge bunch of different ways. The one I talked about today was by taking these 1 megadalton antibody complexes and distilling them down into small macrocyclic peptides that are still able to activate complement, getting rid of all the rest of the antibody, leaving only that ability to activate complement that we can then use like Lego and stick on other molecules as well. And we're now spinning out a company based on this. New super resolution cryoclem workflows, equipment, and software. They really enable structures of intracellular proteins to be solved to sub-nanometer resolution. I'm super jealous of some of the other subtonogram structures I've seen. We tend to go for the floppy, flexible proteins at the moment, but seeing some of the other structures at this meeting has been eye-opening. So I'm looking forward to getting in contact with a bunch of you afterwards. And I just said right at the end, automated segmentation software greatly accelerates structural biology. Now, we are hiring currently multiple postdocs, got 3 postdoc positions open and potentially 2 PhD positions open. So if you're interested in anything I've talked about and you have experience with immunology, coding, light microscopy, building microscopes, cryo-FibSem, please do get in contact. The adverts will be online. One of the adverts is online on Monday and the rest will follow pretty soon. But do please get in contact in advance as well. That just leaves me to thank my wonderful lab. I've only had time to talk about Sebastian Martin's work today. I'd love to take some time and tell you about everyone else's work as well, but some fantastic collaborators in Bristol, the LUMC, and all over the world. The UK and Dutch funding agencies, and of course the EU for awarding me money. Thank you again to the tech team and the organisers for accommodating me online. And thank you all for your attention. I'm obviously happy to take any questions that you have.
Thank you, Thom, for a fantastic talk. And I bet you'll have some emails in your inbox quite soon from applicants. Okay, great. Are there any questions from the audience?
Hi, Thom. I was just wondering if you'd tested small molecule dyes instead of fluorescent proteins for cryofluorescence microscopy.
No is the answer. I've got a collaborator at EMBL, Timo, who's doing this. No, we've stuck with clonable proteins at the moment because that's essentially we can then use, we don't have to reinvent the wheel. We can use existing tagging routines and procedures in our immunological assays. So we've only used green, red, and blue fluorescent proteins. The blue ones we couldn't switch at all. The red ones were only 2 out of the 20 or something that we tested were switching. And obviously I told you about the green ones. But no, we've not done anything with organic dyes at cryogenic temperatures. No, but we are interested in it and collaborating with Timo at EMBL to do it.
Hello, thank you. That was a wonderful talk. I have a bit of a technical question, might be a bit naive, but when your setup, the super resolution cryoclam, is it just like an add-on to the clem, or are you doing super resolution on a different machine? Could you elaborate on that, please?
Yeah, sure. So it's essentially a wide field microscope with some software. And that's it, really. We have a laser box. We built it ourselves from skeletons of other microscopes, kind of borrowed lasers and things like that. It's currently sat in a box. I've moved it from the Netherlands to Bristol. I haven't had a chance to rebuild it yet. All the plans are in that that methods in cell biology paper. There's links to CAD drawings and all the software you need to run it. And do get in contact with me and Mart if you are interested and you run into some problems. But essentially, we built it ourselves, but it's really not that complex at all.
Thank you very much.
Okay, thank you. Let me interject with a question from the Slack channel. Joshua Hope says, great talk. To prevent the blinking of former VAR grids, would it be possible to pre-photo bleach the grid before sample application and freezing?
It seems to be the problem is after vitrification, you essentially remove all of the quenching mechanisms. So things that don't fluoresce or blink or anything at room temperature, because they're being quenched or very short-lived or whatever, all of a sudden become able to become chromophores. So unfortunately, that doesn't help. There's one group in China whose name I've forgotten. Back in 2015, they saw, well, they didn't report it, but I guess they saw the same problem. They got around it by making the FormVar grids incredibly cleanly and then using them immediately. So it is possible to get around it, but it's not possible so far to get around it and have it commercially available, which is really what we're interested in. So we've tried a number of ways of mitigating it, but we haven't found anything yet. And we've tried kind of bleaching it before after vitrification, and it didn't help, unfortunately.
Okay, thank you. Another question from Randy Reed. Inappropriate complement activation presumably does nasty things. Do you know if any pathogens or venomous creatures ever weaponized the concept behind the peptactins?
Yeah, the peptactins. So not behind the peptactins, but there's an horrible cobra venom that activates complement. It's always snakes, isn't it? I think it's probably an Australian snake as well. But yeah, it has this thing that looks a lot like one of the complement components. It can spontaneously activate complement as well. No, things like bacteria, they do the opposite. They coat themselves, they camouflage themselves in complement inhibitors. So one of the things we're trying out is taking one of the complement inhibitors and sticking our peptactin on. In solution, these don't really do anything, but on a surface they can activate complement. So either the bacterium is going to be killed by camouflaging itself in now an activator, or it's going to evolve away from camouflaging itself and then become available to be killed by complement anyway. So hopefully it's early stages, but hopefully it's going to be kind of a resistance immune way of killing bacteria.
Right. We have another question in the audience.
Hello, thank you. One clarification. You said for the super resolution microscopy, you are looking for single molecule localization, like technique PALM and STORM, but RS-EHFP2 is for resolved studies. So I missed the link. Can you explain
It's
more?
essentially it's a very nice photo switchable protein. All of the, well, apart from the structural illumination, I suppose even that does. They rely on pushing proteins into an on and then an off state, whatever that off state might be. So Resolve does it with its triplet states. Other ones do it in various mechanisms. But that protein has been developed to be photo switchable. And it just so happened it was developed by Stefan Jacobs to be used for Resolved. But by virtue of being photo switchable, it's also very useful for single molecule localization microscopy.
Thank you. One more talk in the interest of time from the Slack channel by Katie Downs. Autofluorescence is enhanced at cryo temperatures, particularly in mammalian cells. Do you see blinking of this autofluorescence similar to what you reported with FormVar?
No, not really. It's what background we do see, we tend to subtract. So after activating another subset, we then subtract the immediately preceding view. And this gets rid of kind of a lot of that background. Anything that does turn on then becomes visible. And it's really not the autofluorescent stuff. I know Mart's done some work about this. It's hopefully going to be in the RS tag, the red-green paper as well, where he really shows that it's not a problem. But thankfully, no, because a lot of chromophores become fluorophores when you remove those quenching mechanisms. But thankfully, they don't seem to blink. Yeah.
Great. Let's thank Thom again